So Zelluna is a Norwegian company founded in 2016, and we're developing T-cell receptor-based cellular immunotherapies for a broad range of solid cancers. So we're located in the Oslo Cancer Cluster Incubator, which was the wrong button here, which is very close to the Norwegian Radium Hospital. So our core asset is a portfolio of non-engineered T-cell receptors sourced from long-term surviving patients from cancer vaccine trials. And our current focus is to deliver a T-cell-based autologous product and then moving on to a allogeneic NK cell-based product down the line. Our leading TCR is called ZI-H04, targeting hTERT, and is planned to enter the clinic in the first half of 2020 in patients with different solid tumors. Okay. So I think there are three key elements that differentiates us from the other companies in the field. First one is that we have a unique source of T-cell receptors. As I said, these are sourced from long-term surviving cancer patients from vaccine trials, which means that they have been circulating in a patient for years without showing any signs of toxicity. They are co-receptor independent, which is a sign of high affinity, and they are non-engineered. Second, we're targeting cancer driver antigens such as hTERT, K-Ras, and TGFβRII, which are expressed across a wide range of solid tumors. By targeting cancer driver antigens, you reduce the risk of immune escape and also then of relapse. And the third element is that we are focusing mainly on targeting HLA class 2. Our portfolio also contains TCRs, which are class 1 restricted, but we believe that by targeting the class 2 pathway, we can induce a broad and sustained immune response, which is especially important in the solid tumor setting. Also, class 2 has a much more restricted expression profile than class 1, and is mainly expressed on APCs. So all our targets are essential for cancer growth. If you look at the left panel, or hTERT, this is regarded as a universal cancer antigen expressed in high frequency across various solid tumors. This is the wild-type protein is also minimally expressed in most normal cells, but we're targeting this antigen only with class 2 receptors. The TGFβRII frameshift mutation we're targeting has a more restricted expression profile and is predominantly expressed in MSI-high cancers. This is a neoantigen and is not expressed in normal cells. And K-Ras mutations are also frequently expressed across different solid tumors, and it's estimated that around 20% of all tumors carry K-Ras mutations. So we are targeting the hotspot mutations in position 12 and 13, and these mutations are not found in normal cells either. So as I said, we're targeting class 2, and we believe that we are able to, or by bringing in different immune cells into play, like macrophages, DCs, we're able to generate a much broader immune response. Also by employing different mechanisms of action, we're able to target the tumor cells from different angles. So our product contains of both CD4 and CD8 cells transduced with a TCR. So one interaction point is that you can have activation of DCs, which can cross-prime and activate other naive CD8 cells present. They can also activate other neoantigen-specific T cells in the tumor, which then can contribute to killing of the tumor cells. It's also shown that a lot of tumors express class 2, so you can have direct killing of tumor cells via the class 2 by our product. In addition, you can have activation of macrophages. And by activating these, they can be polarized into a more tumoricidal phenotype and also help killing killing the cancer cells. So we're attacking the tumor cells from various angles. And also this cytokine and chemokine cocktail produced by all these activated immune cells, they will attract new immune cells to the tumor site and then further strengthen and enhance the anti-tumor response. So our lead candidate is a ZI-H04. This is a DP4 restricted TCR recognizing a fragment of hTERT. Now, DP4 is the most frequently expressed class 2 allele and is expressed in around 70% of Caucasians. So if you take this combined with a high expression of the antigen we're targeting, this product has the ability to be used in treatment of a large patient population. I'm just going to show you a little flavor of the data we've generated so far. So if you look at the top left panel here, this shows killing of tumor cells derived from the original patient that the clone was sourced from. If you see the gray, maybe it's a little bit difficult to say, but the gray line here, this is showing killing by our product, which is a lentivirally transduced product. The blue line shows killing by the original patient-derived T-cell clone that our TCR is sourced from. And the red line shows T-cells transfected using mRNA with our TCR. The top right panel shows that our product can actually also prolong survival in a xenograft model, even though a mouse model is not necessarily the best to demonstrate efficacy on a class 2 TCR, which is dependent on other players in the immune system as well. If you look at the lower left plot, this shows peptide sensitivity down to low nanomolar, and the bottom right plot shows an alloreactivity study across 30 donors, which gives you quite a comprehensive class 1 and class 2 coverage, and we're not seeing any alloreactivity. So in addition to developing T-cell-based products, we're also developing allogeneic products based on NK cells. Along the lines of CAR NKs, we are developing TCR NKs. So if you transduce NK cells with CD3 and a T-cell receptor, you get functional expression of the T-cell receptor on the surface of the NK cells, and these cells will kill target cells based on the specificity of the T-cell receptor. This is shown in the plot on the right, where we have tumor spheroids formed by the HCT 116 cell line. And this cell line expresses both the right HLA and also harbors the frame shift mutation that we're targeting. And as you can see on the top panel here, you can see that NK cells transduced with one of our TCRs, recognizing this mutation, is able to kill these tumor cells. And the middle plot shows NK cells transduced with an irrelevant TCR, and this is not able to kill these tumor cells. So our pipeline is gradually building, and the ZI-H04, as I said, is our leading candidate. And in addition to that, we're developing several other TCRs, both for hTERT, TGFβ receptor 2, K-Ras, and we also have several TCR-NK products in development. The ZI-T01, this is our class 1 restricted TCR, recognizing the TGFβ receptor 2 frameshift mutation, and this is currently being used under hospital exemption at the Oslo University Hospital as an mRNA product. So if you look across the TCR-T landscape, we think, or you can see that most other companies is focusing on cancer testis antigens. We are the only ones focusing on driver antigens. It's only Kite Pharma and ourselves that are using vaccinated patients as a source of TCRs. And it's also only Kite and ourselves that are focusing their efforts on class 2. So we think that this puts us in a good position to provide benefit to patients with solid tumors. We're currently setting up GMP manufacturing together with MaSThercell, which is a Belgian CDMO. We aim to have production for clinical trials ready by early 2020 with commercial scalability in the EU and the US in the near future. And to execute on all this, we have a very nice team, and some of the people are shown here. We've also recently hired a CMO, but this is not public yet, so this person is not present on this slide. And with that, I thank you for your attention.